I'm glad to present you NeuroSense Therapeutics. We are a clinical stage biotech company that develops therapies for CNS indications. I'm Alon Ben-Nun, the CEO, and here with me today are Dr. Ferenc Strasik, NeuroSense Chief Medical Officer, our head of the scientific program, Dr. Shiran Zimri, and Nadira Zaltzman, Vice President of Business Development. But first, a disclaimer, since we are a NASDAQ-listed company under the ticker NRSN, and may provide forward-looking statements in this presentation. I'll start with a brief overview about NeuroSense. We were established 5 years ago with one major goal: to provide an effective treatment for ALS patients and other neurodegenerative disorders. Since inception, we have developed a novel drug, PRIME-C, that underwent preclinical studies with outstanding results, leading to the initiation of a clinical phase, a proof of concept phase IIa study that was concluded successfully with promising results. We had a discussion with the FDA, a pre-IND meeting, where it was agreed that this is a 505(b)(2) pathway, which means short time to market. In the short time since our establishment, we have been granted a patent which is valid until 2038 and have additional ones pending, as well as achieving orphan drug designation from both the FDA and EMA, granting market exclusivity of 7 years in the United States and 10 years in Europe. We are also expanding into new indications. We are very excited and optimistic since we found that our flagship drug, PRIME-C, suggests a biological activity in ALS patients. This was shown in a statistically significant manner in a panel of biomarkers that we tested following treatment of people with ALS for a period of 12 months. I'm delighted to present the results today along with my colleagues and to elaborate on the company. We have an innovative approach. While most of the companies in the field are focused on one single target, our approach is a combined therapy to target multiple pertinent targets in a very complex CNS diseases. I'm glad to present to you our devoted, experienced, and diverse team located in Israel, Europe, and the United States, providing widespread coverage in these territories. Mark Luchtenberger, our chair, has vast experience in NASDAQ biotech companies as a chair, CEO, and a board member. Our CMO, Dr. Ferenc Strasik, is a neurologist with vast management background from Biogen, Novartis, and other biotech companies. We are proud to have top-tier leading clinicians on our scientific advisory board who believe in us and support the development of PRIME-C for the benefit of ALS patients.
Here you'll see NeuroSense's pipeline, where we've included 2 other CNS indications, Alzheimer's disease and Parkinson's disease, where we have already begun preclinical testing. In ALS, we have now finished our phase IIa trial, and we plan to begin phase IIb this spring. Now we're going to focus on our ALS program. As you all know, it's a fatal disease that leads to paralysis and eventually death within 2 to 5 years from diagnosis on average. Each and every year, there are over 5,000 people diagnosed with ALS in the US alone, with about 80,000 people living with ALS in the Western world. The burden on the US economically is above $1 billion annually. These numbers, unfortunately, are expecting to grow, as ALS is considered an aging disease and the age of the general population is rising. We expect, based on conservative analysis, that our annual market potential is going to be $3.2 billion.
But ALS is more than just numbers. The person in the photo is Shai Rishoni, who I met with in 2016, when he was completely paralyzed. Shai passed away in May 2018. But before he did, we concluded our first preclinical studies with outstanding results. I came to Shai's home and told him about it, and he was very excited. When I told Shai that now that we understand that we have a therapy to develop, we'll name our drug PRIME-C as a tribute to him. Rishoni is translated to prime in English. This is his legacy.
There is still a huge unmet medical need in ALS. As you might know, there are only 2 FDA-approved drugs currently available. One is riluzole, the other one is edaravone. And very unfortunately, both drugs have only a very mild clinical effect in treating ALS patients. We believe that one of the most important reasons for all these failed trials during the last 15 years is that all these drugs were aiming one single target. We strongly believe that aiming for more than one target, for multiple targets, is heavily needed in order to increase the likelihood to find an efficacious new drug in ALS. And therefore, we developed a new drug consisting of 2 well-established FDA-approved drugs, ciprofloxacin and celecoxib. This new drug is designed to work synergistically on more than one target, on the regulation of microRNA, affecting the iron accumulation and reducing neuroinflammation.
The pathophysiology of ALS is very complex, and it underlies multiple pathways and mechanisms that are involved. And therefore, we believe that the right approach is to tackle multiple fronts. So with PRIME-C, we aim to target dysregulation of microRNA together with iron accumulation and neuroinflammation, all considered key hallmarks of ALS, in which each one of the compounds actually targets several of these pathologies. But importantly, the unique extended release formulation of PRIME-C actually allows us to maximize the synergism between these 2. And so once we had the formulation in our hands, we were able to move forward into the preclinical studies. And this is exactly what we did. So we conducted our studies in Zebrafish model, which considered a very novel and successful model for drug screening. It is highly validated both by the academia and the industry. And so we conducted our studies on 2 key models for ALS. One is the SOD1 mutant and the other one is the TDP43 in 2 leading labs in Israel and in McGill University in Canada. And what we found was really, really promising results. We saw an improvement of 84% in the motor performance of the SOD1 mutant and 110% effect on the TDP43 mutant. And not only that we saw improvement in the motor performance, but we also saw a significant improvement in the morphology, the structure of the neurons. And as they say that a picture worth 1,000 words, so I thought that it will be nice to just see it for yourself. Here we see the motor improvement following the treatment with PRIME-C and the structural improvement in the neurons following our treatment. So in the middle panel, you see the swimming pattern of the zebrafish. We see in the upper panel the healthy ones swimming really nicely, and compared to them, we see the ALS ones really impaired. And following treatment with PRIME-C, we gain a pattern which is very much similar to the healthy ones. And as you can also appreciate from the graph in the left panel, we see the synergistic effect of these 2 compounds and the dramatic effect, especially relative to the standard of care, to riluzole. And this was very, very interesting and actually led us to dive even deeper into this effect and to go and focus on the neuromuscular junction when everything is happening. So here you see in the right panel, we see the presynaptic markers in red and the postsynaptic compounds in green. And when they are colocalized, we see yellow. And as you can appreciate, following treatment with PRIME-C, we gain more yellow, meaning more intact synapses and less often pre and post compounds, which kind of indicate on a healthy neurons that can translate and transfer the signals in a very healthy functional way. And this was also reinforced when we just look on the morphology of the neurons. So here in the lower panel, you see the severe axonopathy with very much kind of look like a Christmas tree branching with the ALS neurons. And compared to that, we see the healthy ones. And Also here we see that following treatment with prime C, we gain a pattern which is very, very much similar to the healthy ones, which actually kind of suggests that there is a neuroprotective effect here to prime C. So with this very encouraging results and the acknowledgement and the support of leading clinicians in the field, we had a green light to move forward into the clinic. And this is exactly what we did. And Ferenc will tell you all about that.
Thank you, Shiran. As Shiran said, based on this very encouraging preclinical data, we conducted a phase 2a study with 15 ALS patients over 12 months dosing. Patients were seen in the clinic every 3 months. Additionally, we did so-called phone visits every 1.5 months. As normal or usual in this phase of development in a phase 2a, safety was the primary outcome parameter by assessing blood tests, by doing blood tests, ECG, and other safety measurements. Of course, we also assessed the efficacy of prime C in ALS patients by using the ALS functional rating scale, which is a global clinical scale, and the forced vital capacity, which is an objective assessment of the respiratory function in ALS patients. And as you might know, the respiratory function is, in many cases, the life-limiting function in ALS patients. And last but not least, we did a huge panel of biomarkers in this study as well. The most important clinical outcomes are the forced vital capacity, as said, an objective assessment of the respiratory function, and the ALS functional rating scale. As you can appreciate, prime C showed a 30% difference versus patients from the PROACT database in the respiratory function. The PROACT database is a huge database consisting of more than 10,000 ALS patients who were enrolled in former ALS clinical trials. So this allows us to do an indirect comparison with our patients treated with prime C. What we did, we handpicked 30 patients out of this huge database who matched the best to our prime C patients according to 9 different clinical criteria. As said, we saw a 30% difference in the respiratory function, but we also saw an 18% difference in the overall clinical assessment of our patients, which equals to almost 2 points in the ALS FRS, which is widely considered as a clinically meaningful difference. In addition of meeting our primary endpoint, which shows that prime C was well tolerated and safe in this study, we could also show a reduced functional and respiratory deterioration, and also very importantly, a significant change in ALS-related biomarkers, and Shiran will introduce these encouraging results to you.
Thank you, Ferenc. So we next wanted to explore the effect of prime C on ALS-related biomarkers, and to do so, we analyzed biomarkers from neuronal-driven exosomes that were extracted from blood samples. So basically, exosomes are vesicles that contain representative content from the brain. They go in spontaneously into the bloodstream and basically allows us to measure parameters from the brain in a non-invasive way. And so we use this very novel model in order to characterize our biomarkers. And to do so, we collaborated firstly with Mass General Hospital, who provided us with blood samples of healthy controls and patients with ALS. And following this establishment of the biomarkers, we next move into the next phase, in which we analyzed markers from our NST-002 study samples from our clinical study. So first thing first, we just, as a first phase, characterize who are the key players in play here. And so as you can see in blue, we turned into our own mode of action and actually established and characterized for the first time markers that relates to a mode of action and relates to the pathology of ALS, and we focused on neuroinflammation and markers that are related to neuroinflammation, and also markers that relates to regulation of microRNA and into iron accumulation. And additionally, we also focused on ALS-related markers, the classical ones. So, of course, we turned to look on the key protein that plays a huge role in ALS, which is TDP-43, and also to two very interesting cellular markers. One relates to autophagy, and the other one is relates to lysosomal trafficking. Both are known to be impaired in ALS and also in other neurodegenerative diseases. So with this cassette of panels of biomarkers, we moved into the analysis. And as you can see here, when comparing patients with ALS to healthy controls, we see a significant change in markers that represent regulation of microRNA and also iron accumulation and neuroinflammation, together with accumulation in TDP-43 and impairment in the cellular processes that I was talking about, the autophagy and the lysosomal trafficking. And importantly, when we treated patients with ALS and looked on these parameters, what we saw that following treatment with PRIME-C, we see attenuation in this basic phenomena. So if we were talking about TDP-43 and we saw the elevated levels of TDP in patients with ALS compared to the healthy ones, and also we know from the literature and formal studies that was conducted that this elevation is also increased with the deterioration of the disease. Here we see that following treatment with PRIME-C, we see a significant reduction over time. And this was very, very encouraging, and actually was also accompanied by the same trend, a decrease in inflammation parameters and the attenuation of the cellular markers that we observed. And so this was very encouraging and actually led us to the next phase, which was to look at the correlation between these very interesting biomarkers and our clinical outcome. So you can see here as one example, we see the changes from baseline in the levels of TDP-43 compared to the functional rating scale, to the ALSFRS-R. And what you can appreciate from this correlation is that in patients in the study in which we were able to lower the levels of TDP-43, we were also able to attenuate the deterioration of the disease, meaning that there are maybe biological activity here to PRIME-C. And this is very, very encouraging. And I can share with you that we also get a huge endorsement by the field following these results.
For the next 2 years, with the funds that we raised, we are advancing PRIME-C clinically, initiating a phase 2b biomarker study in about 2 months. In parallel, we have already started to advance Cogni-C for Alzheimer's disease and Stability-C for Parkinson's disease throughout the preclinical stage, planning to be IND ready by the end of 2022, early 2023. Since the beginning of 2022, we already completed successfully the 21st days of tox study. We submitted an FDA IND on February 16th, so about a month ago, and we started the third stage biomarker study in collaboration with Mass General Hospital in Boston. Apart from the milestones shown here, we expect to generate much more information and news that we will communicate to the public soon. There are 3 major activities that we are focusing on, as you can see in this high-level Gantt. We have a discovery arm to find and develop additional assets that will be the engine for growth of the company and its future pipeline. We are also advancing Alzheimer's and Parkinson's, as I mentioned, to be IND ready by the end of 2022, early 2023, and position ourselves to kick off clinical studies in these important indications. And of course, initiating very soon a phase 2b study in ALS, a biomarker trial. Its results will enable us to fine-tune the phase 3 study design and to significantly increase the success rates. And yet, if the regulatory environment will enable it, we intend to turn this study into a pivotal one, recruiting more patients, and upon successful results, submitting an FDA new drug application to see our ALS drug Prime-C in the market even before 2025.
With a strong patent estate, IP coverage until 2038, and funding to allow our company to reach a critical inflection point, we believe that our company will be able to give hope to ALS patients and others suffering with CNS diseases. A few fun facts about our management team. Not only are we over 70% female in the company, our board is comprised of 50% women. And we also are very proud of the fact that we're international. 66% of our board members are US-based and have vast experience in biotech and in public companies.
I want to thank my colleagues and the audience today. We welcome you to join on our mission to bring new therapies to patients in need. Thank you.